I'm Erica Hayden. I am a reporter with the science journal Nature. I'm very grateful to have been here for this exciting meeting today, so thanks very much for the invitation. So as you know, the topic of our panel is cancer moonshots and extending the reach and impact of immunotherapy. So as we've heard today, there's tremendous enthusiasm about the prospects of immunotherapy. It's a modality that's sent shockwaves throughout the cancer world. Mostly because it appears that some subset of patients go into a lasting remission, a durable remission with this modality, perhaps because they've developed immunity to the original cancer. It's still a subset of patients, as we've heard, who respond to this modality, but we've also heard exciting work that's trying to expand the reach and impact of immunotherapy today. So we've also heard about some efforts that are aiming to raise the prospects of a cure by eliminating cancer through broad-based moonshots. So we've got the National Cancer Moonshot, which was announced by President Obama in his State of the Union speech, in which he pledged to spend $1 billion and led by Vice President Biden. There's also the Cancer Moonshot 2020, led by billionaire Patrick Soon-Shiong, involving pharmaceutical companies, hospitals, and insurers. The question that we want to ask about these moonshots today is, given that the detailed workings of these programs are still a little bit unknown, and given that there may be somewhat of a myopic view of these initiatives, given the potential reach of immunotherapy in other realms, such as infectious disease, what do we know and how far can we go with these initiatives? So my first question for the panel will be, 40 years ago, as we all know, President Nixon declared a war on cancer that has not yet been won. In explaining why now is the time for a moonshot, Vice President Biden said, "The science is ready." Is it?
I can take a stab at that. One of the things that I tried to point out this morning is that the whole concept of checkpoint blockade had nothing to do with the war on cancer, had nothing to do with sequencing the genome program. And yet still, the vast majority, it's kind of hard to see it. I've heard estimates ranging from a low of 3% to even spokesman for the NCI said, no, we're spending about 15% of the money for immunotherapy. But that hasn't changed since the approval of these drugs began. There have been no new initiatives whatsoever. And there's no promise that these new moonshots, I mean, they're talking about it, but you're right, we don't know what they'll be shaped like. I would hope they don't spend the money to enhance even more the RAS program that was just started a year or so ago, where they converted the whole Frederick facility to try to find a way to target RAS, which nobody's been able to touch in 30 years, despite millions and millions of trying. So I think it depends on how the moonshots are there. I think our, and I'm both are saying the same thing, we need to get more people in, and there need to be some special initiatives to deal with the questions that are coming out of the analyses of the patients who are on therapy. We need to know how these things work, and then all the other things. Talked about the right combinations and all this, but it needs to be done properly with the right emphasis. If the emphasis doesn't change, it's going to be the same.
I think we haven't talked about it, but I think Jim was just recently appointed to the board of the Obama moonshot, because it's actually the third moonshot as well as the Ron DePino moonshot.
Yeah. Well, yes.
Why don't we go to a different planet? That's what but
that's if your Trump gets elected.
I'll go to a different planet.
Yeah, Planet 9. Planet 9. Anyway.
A good thing, that was a good thing, but I think, hopefully a very optimistic thing for the future of this particular moonshot. But problem was when it was first conceived, many of us saw very little interest on the part of that entire enterprise in cancer immunology and immunotherapy. In fact, some of us were together at a meeting at the NCI, basically pleading with them to do something. And at first, Biden was supposed to show up at the meeting, then he didn't show up. He was supposed to send somebody, I think, who was there for 10 minutes. And there was a lot of lobbying that was done, I think, by a lot of the professional societies to try and get them in, and I think that yielded Jim, which is a terrific thing. But the problem is that people have been studying oncogenes as the causative agent of cancer for so long. What's developed can best be described as the oncogene industrial complex, where that is such a powerful force in cancer biology that even though everybody else sees what's going on, very few people from that area are really embracing it in a fashion that requires and allows for the transfer of real resources away from what they're doing, away from RAS, and towards the immune system. Nobody wants to give up because everyone is so heavily invested in that area. So there has to be, I think, a fundamental change in the firmament, and the clinical data are just so abundantly clear. It's thrilling to understand, or I just can't understand why that hasn't happened. It's painfully obvious that maybe we're not going to cure everybody, maybe we're not going to cure anybody, but we're certainly doing an awful lot better with immunotherapies than anybody is doing with anything else across such a large array of cancers.
What I'd like to say, and I actually heard this from a person who is a small molecule guy. He said there are more papers on resistance mechanisms than there are patients who've survived longer than 2 years with these drugs. There's 10 different escape mechanisms from the BRAF inhibitors alone. It's an intractable problem approached that way.
Dr. Bardawaj, did you want to say something?
Yeah, I'll just add to what was already said, which I agree with. And I think I'm hopeful, though, with all the inroads that have been made so far, that younger generations, in particular clinical oncology fellows, for example, are going to have to learn how to treat patients, are learning how to treat patients with checkpoint blockade inhibitors. So there is going to be a fundamental change in thinking in terms of approach. There's going to be fundamental changes in understanding how these therapies work because they'll have to know how to do it. So I think we're going to bring up a whole new generation of people, including scientists as well as clinicians, whose mindset will be different from the mindset that we're currently having to deal with now. So I'm hopeful with that, and hopefully some of this $1 billion will get fed into that stream. That we'll see these gradual shifts and change in emphasis on what people working in this field should be doing. And I will say, I think these combinations, and then going back, as Jim and Aaron, or if you're doing to fundamental mechanisms, because cancers are going to be different in terms of how they're dealing with and learning how to manipulate the immune system, whether it's in situ or systemically, and all this. Those are incredibly exciting combinations, and so I'm very hopeful. I mean, I've been in this field for probably longer than some of you were even born, probably, but went from a very tiny little field to something that's growing and expanding. So it's been very encouraging to see that shift.
Another question about language. Oh, do you want to weigh in on that?
Give you an outsider's view. So we've never worked on cancer, still don't. But in the last 2 years, I've been going on cancer after the PD-1 antibodies had such tremendous success in clinical trials. I now go to probably more cancer meetings than any other meetings. And I've been, to me, it's really exhilarating. I went to my first AACR meeting last year, and there were about 25,000 people there. I'd never been to a meeting with more than a few thousand. But more than the number of people was the energy. I mean, the energy was just tremendous. And I had not been to cancer meetings before. And a lot of the excitement was about immunology. So I think it's safe, and I think Ira really alluded to this in his talk. This is an exciting time to be an immunologist. Probably the best time to be an immunologist. You can address truly fundamental questions about the immune system. I'm kind of paraphrasing what Ira said. But also have the possibility of seeing something in the clinic fairly quickly. I mean, I think it's amazing that we are now positioned where we learn, do discovery, and in our lifetimes have the possibility of seeing something. So even for old people like us, it's exciting. But for the young people, I think you can conquer the world if you go into this fantastic area. So I'm optimistic but all the different interesting things that can be done to really tap into the immune system from many different ways. It's also been, I think, very exciting. I had the vaccine center at Emory University, and our focus has been on infectious diseases. And vaccinologists have traditionally focused, of course, on infectious diseases. But I think that people who are trained in that area know about vaccines. I noticed that on Lewis's, at the bottom, he had cancer. Is that right? Didn't you have cancer at the bottom?
We do.
Yes. So I think that it's also an exciting time for people who have been in the vaccine arena, more for infectious diseases, people who have been in adjuvants areas. So I think there's a convergence of immunologists, people in the vaccine arena, people working with adjuvants, to really apply their skills to enhancing immune responses to tumors and cancer. So I'm actually very excited.
Yeah, the only comment I would make is I'm still responding to Margaret Heckler's promise of an AIDS vaccine in 1983, but I will say that the AIDS epidemic really elicited a revolution in human immunology. Prior to AIDS, there was very little interest in human immunology. I remember as a human immunologist back then, I used to have a very hard time getting anybody to listen to me, and of course that's all of the tools that have been used in a lot of these cancer studies were, the memory markers and so forth were developed based on, initially on AIDS money and people going in looking at infectious diseases, first at HIV and then as, Rafi's progressed to yellow fever afterwards as a comparator. So I do think we're ready. I think the explosion in the immune system and in immunology in the last few years is very exciting. And I'm a pathologist. I would have liked to have done cancer immunology, but I didn't. There wasn't any way to do it back then. There was no opportunity. And I think infectious disease and cancer immunology are very closely intertwined. Besides the fact that some cancers can be caused by viruses, I think we really have to hook up and work together and start doing what Rafi's doing, which is, and that we're starting to do is cross-fertilize each other. By the way, we do have CMV vectors that express cancer and mesothelium and PAP and things like that.
Dr. Vance?
Yeah, so maybe have a few other points to add what's already been raised, and certainly what's changed in the last 10 years, and obviously one of those is this increase in understanding of what results in a functional immune response. But I think it's actually been a few other key things that have happened. And one is, and you've heard about a lot of this today, is sort of the increased sort of academic industrial partnership in not only in translational medicine, but one other thing is one of the first things that popped in my mind when I saw Jim last night. It was that I recall a paper that you wrote, I think with Drew, about 2004, and it's really talking about the importance in combinations. But what the problem was is that you had, at the time, very small companies that had maybe, therapy X versus and not therapy Y, and it was difficult for them to get together to actually try a combination in the clinic, which had, a lot of scientific rationale. And I'm guessing this probably rose out at some level as a combination of GVAX and IPI, which of course you had elegant data in the mouse, but it was actually difficult to get the experiment done in the clinic, not because it didn't make sense, it's because the companies wouldn't talk to each other and ultimately combine. And now, with startup companies, and we talked a lot today about neoantigens, from the very beginning, for example, they will combine some of these neoantigen targeted vaccines in combination with immune checkpoint blockade inhibitors. And I think that's one of the fundamental things that have changed actually in translational research that didn't otherwise exist, like 10 years ago.
Excellent. I want to ask a question about the one of the other moonshots, the Patrick Soon-Shiong moonshot. So one of the basic plan for this seems to be to run large sets of clinical trials testing combinations of immuno and chemotherapies. So I wanted to get your thoughts on the value of that approach. Who wants to go first?
But I think that the jumping right into very large trials of combinations without working out the details of the mechanism in small trials doesn't make a hell of a lot of sense. It's going to be, I think, in the ultimate, unless he's got really good reasons for picking combinations, just going to use up patients for nothing. And it's not likely to cause any advances. What these things need to begin with are small, 2 dozen patient trials coupled with mechanistic studies and surrogate endpoints or real endpoints. And, if they do the best combination that makes the most sense, try it. If it doesn't work, move on. The massive thing, I saw him on 60 Minutes where he stood in front of, 75 sequenators or something. I learned later they weren't even plugged in. But I'm not sure. There's a lot to be done that way.
I agree with that entirely, because not all chemos do what I was referring to. Many of them either do nothing or actually do impede immune responses. And I think the large trials we're doing were informed by a lot of work prior to that to know which ones to choose, and we still don't know if they're going to work. And just to do this randomly it just makes no sense for all the reasons that Jim said. And it actually makes less than no sense because probably most of the trials will fail. And then what could have been a very viable and important path forward will just look like just one failure after the next, which is what happened to cancer vaccines early on. People were doing vaccines because they were easy to do without any understanding of how you immunize people, without any understanding about what really the appropriate targets should be. Thousands and thousands of people were immunized or treated with these vaccines. None of them received any benefit. And that whole period, I think, set that part of the field back probably 10 years.
I think with smaller trials too, that are designed to do really in-depth monitoring, whether it's biomarkers, looking at the tissue, looking at the blood, serum, and so forth, and then sequential measures in smaller studies to look at perhaps an initial combination to see if there's a signal, some kind of immune signal that would allow you to move to the next step. We tend to do, progression-free survival, overall survival, et cetera. Maybe we need to step back and really, more closely at the tumor microenvironment.
So one of the points I was trying to make in my last slide, that's been for the last 3 decades, do 1,000 patients, move the median over enough to where you get a p-value. That doesn't, in the end, really help very much.
Somebody who's now increasingly dealing with the commercial side of the business, there's a great tendency and push to hit a home run, to just put something in and have it work, and then have a product and then be done with it. And all these small trials, whether it's in infectious disease and HIV or in cancer, seem to me these are really experimental medicine trials. It's important to have a very good question that is based upon basic science before you go forward, so that you'll answer a question as you move along, and not just go for the home run that this will work or this will not. It doesn't matter if we understand how it works. And the trick is picking what to focus on and what questions to ask. But if you need to have a very well-defined question, I think. Otherwise, you're very likely to waste your time and money.
Other thoughts, Dr. Rahman?
This is maybe going out of my league here to make this, but I think you can. This is a very complex question in terms of clinical trials. When you do clinical trials, how much basic information do you need to know about that combination? And I think it's a question that comes up in vaccine trials, but perhaps even more so in the setting of cancer, because you're dealing with somebody who may not be living for very long. So I don't think it's as easy as saying, well, shouldn't do it unless we really know a lot about it. I would kind of maybe go the middle ground, because I don't think you really know from animal models, tumor models, what really will work. Of course, you learn a lot from it, but I think that a little bit of risk would be fine. educated risk, I think, is probably needed to move the field forward.
I think that the point is educated risk or educated guesses, not just random combinations. It's just not enough.
You have to take some risk.
Yeah, no, because he would never do anything. We are continuously now bombarded with new combinations to do, and there are things where you believe mechanistically there would be synergy. So those are the best ones, but you may be wrong. There are those that may just be additive. So you get some benefit from agent A and some benefit from agent B, but if in the best of all possible worlds, they'll just add up together because they're not going to do anything with each other. And then there are those that will be negative. So A will interfere with B. And so those are the ones that you need to watch out for. And I agree entirely that the predictive power of mice for doing this is very limited, which speaks to why the trials have to be small and have to be done in a biomarker-intensive investigational fashion, that you can then see what's happening and, again, as Jim said very accurately, bring that back to the laboratory and work on it. But if you waited to know everything, you would never do anything in this sphere. So, risk is, speaking as someone from a company doing this all the time, risk is a daily daily diet, but you have to gate it or understand what risks you're taking.
And I think the real challenge for these combinations is the reality of business, because what molecule or what things do you combine is determined by who has the rights for that. So I think that this is, and I don't have the solution for it, but perhaps some of these moonshot or some planet shot could address that issue. That how do we facilitate different companies or different biotech, some way of kind of coming up with the best possible combinations without really, I wouldn't say not worrying about it, but trying to overcome the barriers of different companies working together.
As Tom said, that used to be a barrier, but it's not so bad anymore. For one thing, there's 6 PD-1 antibodies out there.
Right.
Some of the companies are more willing to collaborate, and that's changing for them.
Yeah, which is good. Yeah.
So in the article that Tom is referring to, Drew and I were asking the NCI to please step in
that'd be great.
and help, and they never did actually. So, but anyway, the companies finally changed.
Well, they did. That was the RAID program.
Yeah, that's true.
Just never did anything.
It had about 6 products.
I want to move on to talk a little bit about infectious disease. So we've heard, about the moonshot initiatives in cancer, but I think a lot of people have seen the potential for these approaches in infectious disease. Is the time right for a moonshot to use immunotherapies or vaccines in TB, HIV, malaria?
Yeah, absolutely.
Bring it on.
We've got 3.
Yeah.
And I guess what could be done to distinguish this from, as you mentioned, there's been discussion in the past about HIV vaccines or curing HIV, and it didn't quite get off the ground.
I think that with regard to HIV in particular, I think there's, despite all of the progress that's been made in antiretroviral therapy, there was just a recent study that came out that HIV will not be cured, the HIV epidemic will not be ended by antiretroviral therapy alone, and you're really going to need a preventative vaccine. And if you had a vaccine that was in the mid-2020s that had 70% efficacy, you'd prevent something like 16 million deaths or infections over the next 50 years, and ultimately get down to almost a minuscule amount of HIV infection. So I think there's still a very strongly supported reason for an HIV vaccine. HIV cure is another story. That there's 30, 40 million people out there that could benefit from it. It's going to be a harder wall to scale. But there's been a lot of interest in it. I just came from a meeting where it's a topic of interest, and it's being worked on very hard in a moonshot type fashion. I think it's still getting a lot of support. HIV pathogenesis now, the field has sort of morphed into HIV cure. So all those people are now working on HIV cure. TB is astonishing to me that there's so little investment in TB, and it's such a big problem. It's actually in some ways scarier, multi-drug resistant TB than HIV in terms of what could potentially happen with it. I'm happy to, that the Gates Foundation is taking a big interest in that now, and so that's moving forward. I still think, hepatitis B, a therapeutic vaccine, there's hundreds of millions of people out there with hepatitis B that aren't being helped. I don't think hepatitis C is now supposedly an ended problem, but of course people can get reinfected, and there's no vaccine yet. So I think there is still a huge need to work on the infectious disease side, and I'm really hoping that it's really interesting to me that the HIV cure people are now looking to cancer as their, so all the techniques that have seemed to be working cancer are now being applied to HIV cure. So there's cross-fertilization, there's the adoptive transfer of targeted cells, targeted T cells, there's the anti-PD, the checkpoint blockers, blockades, and so forth are all being looked at in the context of HIV cure. So I think there's a real potential for a lot of cross-fertilization, and it's a very exciting time to be an immunologist in either field.
Yeah, make some. I'll make some comments about some challenges and also some successes. So as Lewis pointed out, the antiretrovirals, which have been around for like a decade now, have greatly impacted the HIV epidemic in terms of changing a death sentence to a chronic disease. But what's interesting is that despite antiretrovirals being there, the number of new infections have remained the same, which is kind of a shocking statistic. Same number of new infections happen in the US today as they were happening 30 years back. So what that means is that we need a vaccine. That's the only way that you will really control the epidemic, is to have a vaccine. The HIV vaccine is probably, within infectious diseases, probably is the toughest target for many reasons. It's the most variable of all viruses. It has very difficult to raise good neutralizing antibody to it. It's one of the most glycosylated envelopes that exist in nature, is the HIV envelope protein. Unless you kind of really stop it in the beginning and control it, like having the right antibody which neutralizes many strains, or some of Lewis's approaches, you will then end up with a latent infection. So the variability, the latency really causes just the intrinsic difficulty in making the right antibody is very challenging. But there have been major progresses in this area. Work done by several groups have identified potential Achilles heels on the HIV glycoprotein, where the region doesn't change much, but antibodies can be generated or are present that will neutralize multiple strains. So this BNAPs is one of the most exciting areas. The challenge there is, how do you induce those? We don't have the answer for that, but I think we have really outstanding scientists now addressing this question. You heard Lewis's exciting data today with this novel vector, which is giving T-cell responses which are very functional and durable. So there is tremendous progress in the HIV field. I wouldn't give a date in terms of when we'll have a vaccine, but I think that there'll be progress. But let me give you a couple of examples of some success. One is the dengue vaccines. Dengue was considered to be one of the most difficult things to control. 4 different serotypes, issues with enhancement. But it's not that we suddenly have a dengue vaccine that confers full protection, but we have something that gives about 30% or so protection. It's not bad. And this is the chimeric vaccine. It's a yellow fever backbone with the envelope of the 4 dengue serotypes. But there's better data coming in the clinical trials from the quadrivalent live attenuated dengue. And these probably will be, wouldn't be surprised if these are licensed in the next few years. So I think there's some interesting development and promising developments towards a dengue vaccine. Also malaria, you can either be critical of what has been reported, or you can say, well, we have something that's 35, 40%, as opposed to having nothing. Malaria kills children. I think people don't realize that. It causes many, brain infections and kills a lot of children. So even if a vaccine that was 30% protective for children would save a lot of lives. So I think there's progress in dengue, a lot more to be made, but there's some promising leads there. Same thing with malaria, two of the biggest ones. Louis mentioned TB. TB is very interesting. The BCG vaccine is given worldwide and does protect against early pulmonary TB. So it is actually effective in the early stage. Where it doesn't work is later on. And there you really need a vaccine, which basically what you need is a booster vaccine. Nobody's ever going to stop giving BCG. That's not going to happen in our lifetime or probably ever. So basically what is needed for TB is really a booster vaccine that will then prevent us from having reactivation later. Okay? So in terms of moons, I think tremendous excitement in the and good promising leads, good challenges. In terms of therapeutics, immunotherapeutics in infectious disease is much harder because of the cost issues and also the balance between side effect, the risk-benefit ratio. But one area which Louis pointed out is hepatitis B. Chronic infections which cause cancer like HCV, HPV, I think there's tremendous opportunity for immunotherapeutics there. Therapeutic vaccines, some blockade with molecules like PD-1 or other strategies. I think that's possible there.
Great. I think I'm cognizant of time. We want to leave some room for audience questions. Is that right? Okay. So I'm going to ask one more question, and then we'll take some audience questions. I wanted to ask what you all think cancer will look like in 10 or 20 years and how a moonshot could change that view.
I think immunotherapy is going to be an integral part of it, of cancer therapy. I think some of the cancers, particularly the ones that have responded well so far, I think we will move to where we can cure the majority of people. I think that there's going to be a lot of really tough ones to still deal with, as I was mentioning, glioblastoma. There have been anecdotal responses, which is promising, glioblastoma and pancreatic cancer, but nothing that really works well. So still a lot of work to do. But I think what you'll find is immunotherapy of one sort or another is going to be in combination with radiation, chemotherapy, small molecules. Vaccines are. But anyway, it's going to change from just one type of therapy to combinations.
I also think we'll intervene earlier. You know, we're ipilimumab has been approved in adjuvant melanoma, and PD-1's being tested. So I think what people refer to as immune prevention for recurrence, that sort of thing, is going to become more mainstream with time. I also think analytics, I always think of the Star Trek kind of, evaluation of the patient comes in and gets a head-to-toe evaluation, and I'd love to see that with the immune system, that we can implement everything we're learning about how to, dissect the tumor microenvironment, dissect the immune response, when a patient walks in the door, sequence the tumor, et cetera, and have a data bank of what should you do for that patient, far beyond what we have currently.
Yeah, hopefully it'll become part of the diagnostic enterprises, as you're saying. Now, there's fascination with sequencing patients and looking for what are generally referred to as actionable mutations, despite the fact that we don't have any drugs that are effective against the actionable mutations. But from those data come also a lot of valuable diagnostics concerning what the immunological status is of a given patient. I think one thing that the moonshot could do, if in fact they can break the hegemony of the oncogene complex, is to devote more big data exercises really devoted to that type of a challenge. We have had an enormous activity with the Tumor Genome Project or Cancer Genome Project and all sorts of genomics. I'd like to see that activity now turned and that energy and innovation turned to analyzing the immune system.
Do we have anything to add beyond that?
Okay. Picker or Dr. Ahmed? Okay. I'll open it up. Are there any questions from the audience? We have one in the back, two in the back here. 3, 4.
Hi, I have actually two questions, one for Ira and the other one for Rafi. The first question for Ira is, are the developments in immune-related therapies for cancer in any way or technologically related to some of the developments we're seeing in psoriasis with biologics? Or are they completely just independent sort of directions? And the second one is for Rafi, is you made the sort of the kind of statement that vaccines are the only way to fight the AIDS epidemic, which I certainly think vaccines are very important and will be a breakthrough. But there was an essay by Tony Fauci about the use of widespread diagnostics in combination with delivery of therapies as an alternate way of looking at how to control the epidemic in the United States. And so I'm wondering, what is the reason why vaccines are essentially, I had quite a question, is it the only way to really solve this epidemic, or are there other ways of doing this?
You want to go first?
I think, if the question is specifically about psoriasis, I think it's really quite different. Generally speaking, immunologics or biotherapeutics that have been used against chronic inflammation have been very successful, particularly in arthritis and inflammatory bowel disease. Psoriasis now is an example of that, where I think trying to turn off or reprogram Th17 cells turns out to be a very good thing to do. Th17 cells, if anything, are probably bad actors in cancers. You might want to get rid of them also, but I don't think will turn out to be quite the lever that they are in a chronic inflammatory disease such as psoriasis. But the idea of using biologics to manipulate specific aspects of the immune response, that lesson has been learned. And I think the early results with the anti-TNFs have been incredibly inspirational, I think, to many.
Yeah, no, let me answer. Yeah. I think you're talking about PrEP. You're talking about PrEP, right? Yeah. So I would agree with Tony that that those things are beneficial, especially when you don't have a vaccine. I think it's something that that certainly should be tried. But I think that approach has major limitations. I'll make a few comments, and I think it'll be good to hear from Lewis also, who's more of an expert in this area. And there are major limitations from that. One is that really the issue of how widely can that be implemented? Just the compliance with doing that, the practicality of doing it. Even with HIV-infected people, you'll be surprised to hear that compliance is actually quite low, shockingly low in some cases, and no more than 20-30% compliance. So I think PrEP will be useful. I think in limited, it's not the solution. It's not the solution for controlling the disease at a global level.
Yeah, I think I referred to, there's a recent publications out, and I can't remember the authors, but basically they took into account the implementation of all non-vaccine prevention methods, and they talked about optimal implementation and what would be expected even with optimal implementation, and they still needed a vaccine to get to, by their mathematical models, to control the epidemic. And that's and even more so if practical things get in the way of optimal implementation, which they almost invariably do. So I think that was a very strong case that a vaccine is still needed. Not to say that these other things don't help, but they're not going to help in the human epidemic with HIV. They're not going to completely on their own in the HIV epidemic.
There's been a lot of focus today on checkpoint inhibitor blockade for obvious reasons, but I was wondering what the panel thought of alternative approaches such as CAR T cells.
Yeah, there's strides being made there. The challenges that are coming up are really finding antigens. In some ways, leukemias are the obvious targets for that because you have antigens that are molecules that are expressed just on one step, and they're not on any other cells in the body. But it's harder and harder to find things like that in most solid tumors. Then there's issues of getting the cells into the tumor. So in the microenvironment, you still have to deal with it. So there's a lot of advances to be made. But right now, there's 50 to 70,000 people have been treated with checkpoint blockades, and at best a few hundred with the CAR T cells and things, despite the fact that they started 10 years earlier. So it's promising, I think.
Next question.
Hi. This is a more down the road, immunotherapy and vaccine therapies. Lots of successes. We hope for these moonshots, but there was already indicated during some of the presentations, scalability, and then eventually also down the road, affordability and accessibility. If we want to cure massively and not only a few individuals, can you comment a little bit on that?
How to make it more accessible? It's accessible now. I mean, with all those things that I put on the slide have been approved by the FDA. Any doctor can prescribe them. And it's expensive as hell. But I think the price is going to come down, particularly as combinations start developing. The development cost of these things was huge, too, because all the rules had to be changed for evaluating responses. They had to have you use overall survival as the criteria, even in melanoma, which is a relatively quick disease. But I think it's going to get better. The main, from what I hear now, the main barrier to a lot of more people getting it is that the adverse events are not as predictable as they are for chemotherapies, where basically everybody's hair falls out. They know what's going to happen. They know how to treat it. And with the checkpoint therapies, some patients don't have any adverse events at all. And then there's a variety of different things can happen. There are good algorithms that have been developed that are in play at all the major cancer centers, but because of the uncertainty and the learning curve, it hasn't really made it into the community settings as it should have. So there's a massive education effort underway by most companies, I think, to try to break through that.
I actually think part of your question is also related to, why I thought we would probably get there when we were discussing vaccines. And of course, there's a huge investment in immunotherapy, I would argue probably for two reasons. One, based on some of the seminal work you heard in the first talk this morning, it works so well. The other point is that it's expensive, and there's a huge amount of, when things are successful that companies can make based on that. And when you think of therapy versus a preventative measure, like, for example, a vaccine, like against TB, when you don't have the unmet medical need in countries like the U.S., then there's frankly less investment and less work on it because there's not support. That's really, I think, one of the foundational problems in development of vaccines. Harvesting all this knowledge that now exists, and it's just you don't see the same extent of work in those fields.
Okay, I think we have two more questions. 1, 2.
So I'm just curious about as you mentioned about the immunotherapy. So as we just entered into the field, I was curious about the toxicology studies, and also what are the some of the short-term versus long-term undesired immune response from these therapeutics.
The toxicology aspects of it, they're different. And as Jim was saying, they're complex. Even with a single agent, there's a range of different types of adverse events and complications that can arise in patient to patient. Some of them may be unmanageable. The vast majority of them, I think, though, are manageable. And they look scarier now than they really are. If you go back to the early days of chemotherapy in the 1950s, people, mostly kids in those days who were being treated, were dying right and left as a consequence of being dosed with very toxic agents and regimens. This field is starting off in a much better place than that. There have been very, very few examples of people actually having such bad adverse events that they die from the therapies. Instead, you seem also to have a situation where if you can reset the immune response, you can either reduce or in some cases even cease therapy, even if there's still a tumor present. So there's just not enough, even though thousands of patients have now been treated, tens of thousands, I don't think there's still enough, there's not a large enough, as we say, safety database to really understand how these things are going to play out in the future.
One of the surprising things, too, is that in the early trials, people with existing active autoimmune diseases were basically excluded from the trials. But since with the approvals, that's no longer the case. And there's probably a lot of people with autoimmunity that are being treated, but yet it doesn't seem to be a big problem.
Or people with brain metastases were also routinely excluded from clinical trials. But it turns out that they're responding, including President Carter.
Last question.
Test, yeah. Increasingly, NIH panels and journals are demanding human data and translational data for publication. This seems is associated with some controversy over the utility of animal models for predicting successful disease research. But ironically, I find in my interactions with companies that there's a more keen interest in these animal models than you sometimes get from journals in the NIH. And I think that's really kind of telling. And I personally find the inflexibility of these policies to really be stifling to progress. And when you think about it in the context of moonshots, it's as if, you know, we're going to sort of go with the technology we have. We're going to not be interested in developing new technology because the new technology is generally coming up from animal research, and I think the advances in cancer immunotherapy are a poster child for that, right? They came up from animal research, and they've translated beautifully into humans. I'm just curious whether anyone has any comment on that.
I think that the pendulum first swung from the transplantable models and stuff to the genetically engineered models because, you know, the oncogene people felt that, you know, that they could more accurately put together, you know, and test drugs, and that may be true. I'm not sure it's even true for the small molecules, but for immunological studies, they're essentially worthless because, you know, they only have the tissue-specific antigens generally, which aren't very good, so they really can't be modeled. I think Ira and I talked about this one time, but they're still extremely useful if you use them the right way. You know which each model, not from what tissue it is, but what its microenvironment is like, and you know how infiltrated it is, what's going on. But you can't say, oh, this is a good model for melanoma and this is a good model for kidney cancer, because that doesn't translate. But they're still very valuable, and I don't know why. I mean, I had a paper rejected, and they said I was using antiquated animal models, and I should be using GEM models, and they were the same models that got CTLA-4 into the clinic, you know, but the next paper on ICOS was rejected because we hadn't moved it into the more modern animal models yet. And so, but there's got to still be a place for it. I think that is going to be a battle we're going to have to fight, though.
Yeah, I think the mouse models do two things. One is you can discover new biology in a mouse model that you're not going to do in patients. And also you can, as I think many of us have been saying, you can take hypotheses that are generated as a consequence of doing work in patients that you cannot do, causality type studies with, and then take those back into the lab and into mouse models. Again, being completely agnostic as to, is it a colon cancer model, is it this? But just, look for a model that recapitulates the immunology that you were observing in the clinical trial. What mice don't do, and I think this is where people are stuck on this, is they don't predict response very well. And in fact, I'd argue even in conventional drug development, they don't predict response very well either. The GEMs have proved to be completely useless, even for small molecule inhibitors, because they're too expensive and the throughput is too low. And just normal xenograft models have been as predictive, if not more so, or looking at normal small molecule type inhibitors. So I don't even think there's anything as peculiar to immunotherapy. That's just a deficit of mice are not human cancer patients, but that doesn't mean they don't have an absolutely critical role. Maybe the pendulum has swung too far in the wrong direction in that regard, and perhaps as a consequence of discussions that would occur as a result of the moonshot, the NIH and the NCI will be instructed to not be quite so dogmatic.
I think one relevant point, beyond that, and particularly I think this is a case for, first in human agents where, again, acknowledging that, the responses in mice may be very different for a number of reasons, just, receptors expressed in different tissues, everything that you know about. But I think it's also very important from the regulators. So the FDA, in many cases likes to see, some analogy of what the expectation is for efficacy in humans, and I think in those particular instances, responses in particular mouse models can be very helpful for sort of guiding the FDA through the process of what the rationale is for translating a particular drug.
I'm glad you brought up this question about publications and needing human data. And I think we need to push back. I think it's partly a trap we've created for ourselves because we are the reviewers, and even we are guilty at times of somebody has a very nice paper they've submitted to Nature or Science high-profile journal, and then we say, what some reviewers say, and then the editors insist on some human data. I think it's a bit of a disservice because, and then what often happens is that some little figure is stuck at the end, which is a very preliminary study, because obviously you're not prepared to do a full-fledged study. I think as scientists we should push back on that. Certainly you want extension of that in humans, but that really is a different study. It's not figure number 5 or supplementary figure number 26 of your existing papers.
All right, well, you will have the last word. So let's heed Dr. Ahmed's advice. Go forth and conquer the world. Thank all our speakers for today. Thank the organizers.
Thanks, and thanks to everybody here. I hope we leave with an appreciation for the importance of immunology in cancer and infectious disease. I want to thank all the people who made today possible, and first and foremost our speakers, but also the faculty team who helped invite everybody, David Roulet, Laurent Coscoy, Russell Vance, Dan Portnoy, and Jeff Cox, and also to Zalose Lyons and Annie Lynn. Zalose is out in the back and has been making this thing happen all day today, and of course all of our excellent volunteers. Thanks to all of you. Please visit our websites, send.berkeley.edu, crl.berkeley.edu, ivry.berkeley.edu. Please return your surveys outside in the boxes provided, and also you can recycle your name tags in boxes on the table outside as well. And please join us for Bay Viro if you're into virology on May 27th. And of course, next January, the 9th Sen Symposium will be back at its regular time. So we hope to see you all then. And for now, please enjoy the reception and the posters outside. Thank you.